So welcome to join us for today's webinar. Today's topic will be talking about scanning ion conductance microscopy, SICM. And let me give you a brief introduction to our presenter today. So today's webinar will be presented by Dr. Jae Kim from Park System, Korea. Dr. Kim received PhD degree in Material Science and Engineering from Nanyang Technological University, Singapore. He joined Tokyo Institute of Technology International Research Opportunity Program in 2014 and was engaged in nanoscale chemical composition analysis using scanning probe microscopy. So from December 2017, he started to work in Park System as the Manager of Application Technology Center. He specialized in the enhancement of nanomechanical measurement performance for atomic force microscope, building up an accurate and reliable environment for electrochemical measurement using scanning probe microscopes, and scanning ion conductance microscopy application development for biology. All right, so without further ado, I will share the presentation from Dr. Jae Kim. So just for information, if let's say you have any questions during the presentation, feel free to type your question in the chat box. We will bring this question up to Dr. Jae Kim, and he will answer your question right after the webinar.
Hello everyone, this is Jae Kim from Park Systems. Today I'm going to talk about Park Systems' very interesting measurement mode, the scanning ion conductance microscope, and we can call it SICM. Generally, the scanning probe microscope, or AFM, is utilized very tiny and lazy tip for monitoring sample surface. But in case of SICM, the glass nanopipette is used as the probe for imaging the sample surface. And it is very specially designed technique for biological sample in liquid condition. Okay, now let's take a look at what SICM is based on principles and applications. Since AFM is developed, various applications of nanotechnology were established, and those applications have led technological advances in many science fields. Using X, Y, and G scanner, three-dimensional information of sample surface can be constructed. A few nanometer size tip scans the sample surface by using interaction like atomic force between AFM tip and sample surface, like this. As shown in these images, researchers can acquire very detailed topography of nanostructures and accurate probe positioning and distance control in nanometer resolution. Based on those benefits from AFM's very practical imaging capability, various applications were developed and enabled many innovative material property characterizations. And it directly has led to advances in research and industry with very successful prosperity. Together with the topographical information, electrical, mechanical, magnetic, thermal, and other properties can be investigated using AFM. Like this, in this slide, in electrical properties, we can measure the localized charge or surface potential, and also we can measure the current, piezoelectric resistance, and other electrical properties can be investigated. And we can also measure the modulus, adhesion value, and like this, the relative mechanical property or hardness and lateral force can be investigated. And other properties, of course, we can measure by AFM. But however, those technological advances by nanotechnology has limited in ambient condition. very soft samples, like biological samples in liquid environment, there is not much successful advances with high impact. Because of unexpected physical contact of AFM tip on sample surface, small and fragile structures can be dragged or scratched. Also, AFM tip directly affects to samples features, and this may change the condition of the lifestyle. Of course, this example may be the condition of an actual lifestyle, but AFM tip may have accelerated the breakage of the cell's intercellular bridge. Well, to be a practical in-liquid imaging tool for biological sample, it seems that the tool should satisfy the following three requirements. At first, it should be acquire images in true non-contact. As second, it needs to have stable imaging feedback during imaging. And lastly, the imaging process, the imaging operation must be simple and easy. Fortunately, the Park Systems SICM meets these requirements, these three requirements. SICM is developed for topographical imaging of biological samples in conducting solution, normally physiological buffer. And it allows scanning of the membrane of single live or fixed cell non-invasively. And it enables measurement of cellular activities. The main difference between AFM and SICM is the probe and feedback system. In AFM, very sharp and rigid tip is used, and normally the tip radius is around 10 nanometer or less. But in SICM, the glass nanopipette is used, and the inner diameter of this nanopipette is around 50 or 100 nanometer. For the feedback system, the atomic force between AFM tip and sample surface is utilized for AFM imaging, but in SICM, the current flow between the current ion current flow between nanopipette and sample surface is used. These differences make SICM a better technique for biological sample. As shown in this figure, of course, AFM can image the cell samples or biological samples, but the imaging quality is not guaranteed. But on the other hand, the SICM can image even the softest cells, like a stem cell or neuron cell. Okay, let's take a look at this comparison images of AFM and SICM applied to the actual fibroblast cells. Let us confirm that the SICM represent even very small features on the sample surface very well, like this. But AFM shows these features, very small features, just small pictures by crossing them, like this. Also, in addition, in AFM image, the sample surface appears to be pressed by AFM tip and sample surface interaction. And also you can see this actual line profile. It shows clear difference between AFM and SICM. In SICM, this red line, it shows around 10 micrometer of cell height, but in AFM, just 5 or 6 micrometer of cell height. It means AFM tip pressed sample surface, and this kind of strength of cell height is represented. Okay, then from this slide, let's see why SICM is a good technique for biological samples and what factors enable distortion-free imaging of cell samples. SICM uses a glass nanopipette as a sensitive probe based on electrode inside. The nanopipette, the inner diameter of nanopipette is typically around 100 nanometer, and two electrodes introduced. One is the pipette electrode and the other one is base electrode. And both electrodes, this pipette and base electrode, are made of silver chloride. The pipette electrode is inserted inside of the nanopipette, while the sample is placed in a petri dish that is typically filled with the same electrolyte. And base electrode is placed in the petri dish. In this setup, if we apply the voltage, it becomes some kind of electrical circuit. By applying a bias voltage between these both electrodes, the ion current can be detected, and using this ion current, the locally resolved imaging of samples topography can be obtained. As I said, SICM is a very suitable technique for biological sample due to its true non-contact mechanism. And here is the reason why it is a true non-contact. SICM normally used ion current, which is generated in electrolyte and flows through the pipette tip end. And it means the SICM detects the amount of ion current signal at the pipette tip end, and when the nanopipette is very far from the sample surface, the ion current is mainly determined by the resistance of just nanopipette. But if the distance between pipette and sample surface decreases, the ion current begins to be affected by the excess resistance of the nanopipette and starts to decrease. Detecting the ion current signal, SICM can operate it in accurate feedback without pipette to sample surface contact. During imaging, the ion current signal is always kept maintaining about just only 1 or 2% current reduced distance by feedback system. SICM has two imaging modes. One is the direct current, or we can call the DC mode, and the other one is the approach, retract, scan mode, and also we can call ALS mode. In DC mode, the system keeps constant distance between the nanopipette and the sample surface. After approaching, the nanopipette moves pixel to pixel while maintaining a constant distance from the sample surface. And it is just like a raster scan. And the G position of the nanopipette end indicates the morphology of sample surface. But one critical point of DC mode is if the sample has a steep slope or like a complicated structure, the nanopipette cannot detect this structure and it may damage the nanopipette or sample surface. So I can say it is not suitable for some biological sample or complicated features. The alternative one is the ALS mode. It is specially designed for cell samples. The nanopipette approach and confirm the sample position, and it is retracted by the input control height. And it moves to the next pixel and approached. Means approach, retract, and go to the next step. It is just one cycle, and this process is repeated while the scanning. Of course, the ALS, the scan speed of ALS is slower than DC mode, but it can measure the complex or complicated features of sample surface without distorting or damaging the sample surface or pipette. Okay, let's take a look at more deeply about ALS mode. Here in this position, the pipette and sample surface are very far from each other. And of course, in this position and in this area, there is no interaction between pipette and sample surface. And also, of course, there is no ion current change. But the ion current, the current value decreases as the pipette gets closer to the sample surface. Close, 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 then the ion current decreases. And here we can confirm the distance between pipette and sample surface by designating a specific current value. And this is called approach. And after approaching, the system records the position of this scanner and retract like this. After retracting, the pipette moves to the next pixel and approach. This approach and retract at each point of image repeatedly. In this way, Park Systems SICM shows excellent capability without any disturbance in liquid environment, even for soft cell imaging in liquid. Okay, next is the real view. This is captured image, how Park SICM operation is done with its software, the Smart Scan. On the left, here, the nanopipette and 2 electrodes are set up and the sample is placed on the XY scanner. And using this optical microscope, this pipette tip end and sample position can be monitored. Also, using current log, the current signal tracking is available. This is the example images of a fixed cell by SICM. As you can see these figures, intercellular bridge and nucleus and other some cell structures are available to be monitored. And identifying a specific position of very highly dense neuron cell area next to the left brain tissue, SICM can acquire the complicated morphology in high magnification and without single contact to sample surface. As you can see on the right side, in SICM, the cells reach their long arms to the other cells. So they connected each other and forming extremely complicated network structure. But using SICM, we can image and we can monitor this kind of very complicated structure and cell sample surface. Of course, SICM can apply not only fixed cell, but also live cell. These images indicate the time-lapse imaging of the cell differentiation process of live cells. It is possible to observe in detail the time-dependent movement of how the protrusion at the end of the cell membrane move for cell differentiation and binding. Next, let me introduce some interesting application in terms of physiological imaging by SICM. In this experiment, we prepared the Q7 cell, which is human liver cell line, and the main goal of this application is to investigate the cell membrane changes by detergent treatment. Means how detergent affect the cell membrane, like this. And we used the 1% Triton X100 for detergent and ARS mode, the SICM ARS mode with the 100 nanometer nanopipette were used for SICM imaging. The main role of detergent on the cell membrane is disruption the cell membrane and the formation of very tiny holes in the cell membrane surface. Through these tiny holes, the internal cell membrane proteins can be extracted while the external antibodies can be introduced. And SICM successfully investigates these tiny holes on cell membrane. Compared the fresh cell, no detergent treatment has quite very smooth cell membrane. But after detergent treatment, rough membrane with the lots of tiny holes can be imaged. And approximately around 1 micrometer size holes are displayed and confirmed in this line profile. And if you look at the enlarged image, you can see more detailed cell membrane changes. And as in these images, from this kind of coarse hole to very fine and tiny holes are investigated. All images can be checked in 3D, and the size of detailed features in the line profile can also be checked. And visualizing such kind of very soft and complicated cells membrane is possible with the Conductance SICM. Next one is the cell study by AFM and SICM. In this application, cell surface was imaged by SICM and mechanical properties were measured by FD spectroscopy of AFM, the first distance spectroscopy. And also ID, the ion current distance spectroscopy of SICM was measured. Especially the goal of this study is cell surface image and mechanical property measurement as a function of fixation concentration. We applied the paraformaldehyde called PFA as a fixation agent, and concentration was 10 to the negative 5 to 10%. First of all, we compared the morphology of the live and the fixed cell by SICM ARS mode. Of course, there is no clear difference between two cells, live and fixed cell, but in case of fixed cell, it is slightly shrunk so that the cell adhesive area and the cell volume were lower than those of the untreated cell, like a live cell. And it is good agreement with the anecdotal observation in biologic field, and also slightly raised features are identified, which are expected to be changed in membrane during the fixation process. And then, to investigate the mechanical properties of cells, first spectroscopy by AFM was conducted. From the first spectroscopy, we can obtain the force distance curve, this one, and using force distance curve, we can measure or we can calculate the stiffness, Young's modulus, and adhesion properties like adhesion force or adhesion energy. To investigate the effect of PFA treatment on cell surface stiffness, the first spectroscopy was performed on live and fixed cell, in this case PFA-treated cells. The left figure, this one, shows the typical FD curve for live cells, fixed cells, and solid substrate, and in this case Petri dish. Clearly, the fixed cell shows a much steeper first curve slope. This red one is the fixed cell, and this black one is live cell. And this first curve slope shows the fixed cell is harder than live cell. And the light figure, this one, shows the average Young's modulus value as a function of the concentration of PFA. Compared to the live cell, here, live cell, the stiffness of PFA-treated cell gradually increased with higher concentration. And it means that the PFA treatment affects the cross-linking of cell surface proteins, including affecting filaments. And from this result, we concluded that the stiffening of cells directly results in the cross-linking of proteins. Together with the FD, like a force spectroscopy, the cell surface fluctuation tests were conducted by ion current distance spectroscopy. We can call the ID curve or ID spectroscopy. Paying attention to the fact that the ion current value changes according to the distance between the pipette and sample surface, and the ID curve was measured in each condition, and the cell surface fluctuation were calculated. Here is the result of ID curve. In the solid substrate, like a Petri dish, the ID curve exhibits the steepest slope, this black one. And in case of live cell, it means that the untreated condition, the ID curve shows the broadest slope, this one. Of course, in the PFA treated cells, the ID curve exists in between this region. And these all ID curves were fitted, and the RMS displacement of cell surface fluctuations on treatment of different concentration of PFA are shown in this light figure. As SICM images, the chemical fixation caused no difference on cell surface topography, but the surface dynamics was drastically varied. It means that this live cell had more active movement compared to the PFA treated cell. Smaller surface fluctuation in PFA treated cell can be explained by the PFA treatment effect that result in cross-linking of proteins between the membrane and cytoplasmic proteins. Okay, here is this one is the last slide of today's presentation. To summarize today's talk, the SICM is the technique which enables the biological samples in liquid condition with high resolution and high quality. Live, fixed cell, and other biological samples can be investigated without any distortion. Unlike AFM, which used a very sharp and rigid tip, SICM used a nano pipette with inner diameter of around 100 nanometer as a probe. Since the ion current flow between the pipette and the sample is used as a feedback, it can be measured only in electrolyte, that is, in liquid condition. Together with the other techniques, biological samples can be analyzed from various viewpoints. Thank you for your attention. Thank you.
Great. Thank you very much, Dr. Kim, for the presentation. So, to the audience and attendees to this webinar, so if you have any questions, please feel free to type in the chat box, and we'll go through together later with Dr. Kim. All right. So, I think we have not received any question yet. But Dr. Kim, I have one quick questions regarding the SICM measurement. Regarding the FD measurement by using the SICM method, so are we using the nano pipette to physically probe to the cell surface to get the FD curve? Is that how it works?
Well, no, actually that
we use
FD
the
curve
AFM
is
probe.
just using by AFM, the AFM tip, because
I see.
AFM, the principle of AFM is utilize the atomic force as a layer
Yep.
force between the tip and sample, so we can measure some physical or mechanical properties. But as SICM case, if the pipette contact to the sample surface, actually we cannot get or detect the current signal. It means we cannot get any signals from the pipette or system. So if you want to measure the mechanical property like modulus or some other agent something, you have to use the AFM. But as SICM, we can apply the ID curve, ion current versus the distance curve. It means we can measure some surface fluctuation or surface activity of the cell or some biological samples. Yeah, this is my answer.
All right. Thank you, Dr. Kim.
Mm-hmm. You're welcome.
I think we don't have any other questions today.
Uh-huh.
Yep. So we will end the session today. Thank you very much, Dr. Kim. And
Okay, thank you very much. Yeah, thank you for your support.
have a good day, everyone. Thank you and bye-bye.